to the organizers for inviting me to present my work, and thank you everyone else for being here today. My name is Jordi Abante, and I have been a member of the Goodseals Lab at Johns Hopkins University for the past 5 years. Today, I will be talking about some of our latest work on epigenetic energy landscapes and estimation of these landscapes from nanopore sequencing data. Epigenetics is the branch of science that studies the set of chemical, heritable, and often reversible marks that regulate the central dogma. This figure illustrates different epigenetic mechanisms that cells leverage to function. There are many different types of epigenetic marks, and today I will focus on DNA methylation, in particular 5-MC DNA methylation in Sinji context. Studying these marks allows us to fill in the gaps in the central dogma of molecular biology and to answer many relevant questions, since epigenetic marks happen to play critical roles in a number of health endpoints, for example, in cancer. How do we go about studying these marks, though? Let me briefly discuss that in the context of DNA methylation. Traditional bisulfite sequencing has been the go-to technology for comprehensive methylation studies. Briefly put, the bisulfite treatment and subsequent PCR transform unmethylated cytosines into thymines. After aligning the reads, we can determine the methylation state based on these modifications. Without going too much into the detail, this technology only allows to study 5-MC DNA methylation and produces very short reads, around 150 base pairs. Nanopore sequencing, instead, offers an exciting paradigm that overcomes the main culprits of bisulfite sequencing. This technology can produce very long reads and simultaneously contains information about several epigenetic marks. This gives nanopore sequencing an incredible edge over bisulfite sequencing in certain applications, such as the study of repetitive elements or in allele-specific analysis, for example. But it also enables a new set of analysis that can be done jointly over multiple epigenetic marks. Nanopore sequencing, however, has an important limitation, which is that the current signal is rather noisy, and that needs to be taken into account when performing the downstream analysis. To that end, we built upon two previous works. On the one hand, we used the DNA methylation landscape proposed in 2017 by Jenkinson et al., consisting of a biology-inspired landscape energy function, which assigns a potential energy to each possible methylation pattern. High energy values indicate improbable methylation states, and vice versa. The main idea can be seen in the left figure. This model will constitute the hidden, unobserved state in the proposed approach. But how do we account for the noise in the signal? Well, we do that through Gaussian emission probabilities previously estimated by Simpson et al. So we can combine these two ideas to create a hidden Markov model where the hidden layer consists of the DNA methylation landscape, represented by the random vector X, and the observable layer consisting of the current signal Y. Now, to estimate the parameters of the latent methylation landscape model, which is our ultimate goal, and we're going to call theta, we can use the expectation maximization algorithm. As many of you know, the EM algorithm consists of alternating an expectation step and a maximization step, like shown here, until convergence. This process involves many computationally heavy operations, such as computing the partition function of the methylation landscape several times, and this can be a hard hurdle to overcome if done naively. To overcome this issue, we leverage the transfer matrix method, which allows us to compute the required quantities rather efficiently. There are a lot of details that I'm skipping, so please ask me later if you're interested. In any case, to study the validity of our approach, we performed realistic simulations using Deep Simulator, a recently proposed method to simulate nanopore sequencing. So using simulation, we studied the performance of the approach in estimating the expected values of methylation frequencies, and we compared our approach to two models that binarize the nanopore data by applying a simple threshold on the log odds of the conditional probabilities computed by nanopolish. On the one hand, we looked at a naive empirical estimator, which is what most researchers are currently using, here shown in orange. And on the other hand, we looked also at a model that consists only of the hidden layer of the HMM that I showed, and it doesn't model the noise. And here it's shown in blue. Overall, we found that modeling the noise leads to substantial gains for the various noise levels, and including the real noise levels. Here I'm showing the estimation error, so the lower the better. And we can see that our approach, shown in green here, performs consistently better, and it's more robust to higher levels of noise. We also studied the behavior in estimating higher order moments, such as correlations between contiguous CPG sites and other quantities such as methylation entropy, for example. And we found that our proposed approach is superior across the board. So here we presented the first method for landscape analysis for nanopore sequencing data. We showed that modeling the noise in the current leads to substantial gains, and we propose an algorithm that is robust to various degrees of noise, making it very general. This allows for high levels of noise in the data, which means that the sequencing could be done faster, for example. Our approach extracts more information from the data, and it provides significant savings in terms of sequencing cost and time per read used in the downstream analysis. This has huge implications considering that sequencing takes days and thousands of dollars. This work also lays out the foundation for the study of other epigenetic marks or even epitranscriptomic marks, and to study, for example, the interplay between chromatin organization and the epigenetic landscape. We have implemented all of these in a Julia package, together with a versatile differential analysis method, which will be made public as soon as the full paper, which is currently under review, comes out. In conclusion, we believe that the proposed approach can enable and accelerate both basic and clinical research. With that, I would like to thank the other authors of the paper, the collaborators, and the funding agencies. Thank you very much.